everyone, this is the notes for oxidative phosphorylation. We are using the back of the mega sheet, the last two squares on our mega sheet for unit 3, and I'm going to use the fold in our paper as an imaginary divider for the two processes we are going to be talking about to create ATP. So in this process, and I am doing this landscape, so I folded this hamburger style for those of you following along. In this process, we are going to make ATP. So we already talked about substrate level phosphorylation in the process of glycolysis and the Krebs cycle. So this is now the second way to create ATP, and this process is called oxidative phosphorylation. So this is, at the top, I'm just going to put the title of the processes that we are doing. So it's oxidative phosphorylation by an electron transport chain. So there are two separate processes that we are going to talk about, and I'm going to use this divider to talk about what these two processes are. So your oxidative phosphorylation, the purpose of this is to create ATP. We're not going to do this by substrate level phosphorylation. This is a different process. And we're still going to be phosphorylating ADP, but the energy we're going to get from it is going to come from something known as the proton motive force. Now, in order to get that energy for the proton motive force, that is the role of the electron transport chain. We are going to create a proton difference, a proton concentration difference. So a hydrogen concentration difference. And remember, because hydrogen is polar, see that positive charge right there? Because hydrogen is polar, it's not going to be able to cross the phospholipid bilayer due to the tails, the hydrophobic tails in the bilayer. So we're going to talk about both processes across these, but we're actually going to divide it first into two different sections. First, we need to create that chemoosmotic gradient, in other words, the difference in hydrogen concentration. Then we can talk about the ATP being produced. So what I'm going to do is I'm going to draw a very simplified version of your phospholipid bilayer. This is the one that's going to be the inner membrane of the mitochondria. And then down here at the bottom, this is going to be the space inside the inner membrane. This is the matrix. Over here, I'm going to draw a even more simplified, just as a boundary, that outer membrane. And then this in here would be the inner membrane space. And then so out here would be the cytoplasm at the very top. Here's my outer membrane, the host membrane, and then the inner membrane of the mitochondria. Remember that this is all folded up into cristae, and then you have your matrix. So what I'm going to do is I'm going to take these two processes and talk about them. And the first thing that I'm going to do is I'm going to talk about the electron transport chain to create that difference. We're going to do that on the left side of your paper. So I'm just going to go ahead and fold this just so that I can zoom in and so that you guys can see what some of the stuff that I'm talking about. So an electron transport chain is thanks to transmembrane proteins that are embedded in that inner mitochondrial layer. So you have protein complex 1, which is your NADH dehydrogenase. You have protein complex 2 and 3. We're just going to draw those together, kind of simplify this process. And that's 3. And then we're going to draw the last one, which is 4. So your first protein complex, these are all enzymes, and they're all enzymes embedded in that inner membrane. They are all given specific names, so that's NADH dehydrogenase. Your protein complex 3 is the cytochrome C reductase, and then the fourth protein complex is the oxidase. So what these do is these act as pumps to move hydrogen across the membrane from the matrix to the inner membrane space. We'll put M for matrix and then this for inner membrane space. Now, they're not just open gates that will allow hydrogen to flow through. They're going to pump them across, but they're not open all the time. They will only be opened when they have an electron present. And the electron is going to get donated by the coenzyme NADH, which is now oxidized down to NAD+. So this NADH gives up the electron to this first complex, which is called NADH dehydrogenase. And now that NADH dehydrogenase receives the electron, think the electron as a key that will unlock this pump and allow for a hydrogen to move across the membrane. Now there are hydrogens down here in the bottom part. I'm just going to draw a couple, not too many, because we want to create a higher concentration of hydrogen out here. This NAD plus is now free to go back and pick up more electrons, either in glycolysis, in the oxidation of pyruvate, or in the Krebs cycle. So this is the same one that we've been talking about for the past few days in class. It's that same coenzyme that's going to go pick up more electrons and then come back and keep delivering them to this first protein complex, NADH dehydrogenase. Once the hydrogen has been pumped across, the electron is going to get transported to the next protein complex, and that transporter is called ubiquinone. It's going to ship it to the next protein complex, and when this one receives the electron, it also is going to pump a hydrogen across the membrane. Then the electron is going to get shipped by another molecule to the last protein complex. This is our this is our cytochrome C oxidase. So cytochrome C is the electron carrier that's going to take the electron from this one to this one. Think of it like FedEx or UPS shipping a package. This last complex, which is your cytochrome C oxidase, it's got two jobs. It's going to multitask. Not only is it going to pump a hydrogen across the membrane, it will also take that electron and donate it to hydrogen floating in the matrix and oxygen to create our final product, which is going to be water. So oxygen is what is known as your final electron acceptor. And that's why you breathe in oxygen, so that cytochrome C oxidase can donate that electron somewhere instead of just leaving it lying around. We're going to donate it to oxygen and form water in the process. So for every NADH molecule that gives up the electrons that it picked up in one of these processes, we are going to pump in through these proton pumps a series of hydrogens, or what are called protons, across this membrane. And this area starts to get very packed full of hydrogens. The hydrogen ion concentration gradient gets very high, which means that it would have a lower pH than in the matrix. And this is the job of that electron transport chain, is to create that difference in the hydrogen ion concentration gradient. Now we're going to move on to your oxidative phosphorylation part, which is we're going to do over here. This is where we're going to generate the ATP. And ATP is generated by a special enzyme called ATP synthase. And ATP synthase is like a wind turbine that moves, and as it spins, it'll take some of the energy as it's spinning and use it to take ADP and phosphorylate it into ATP. Now, the reason why it spins is because hydrogen is going to move down its concentration gradient back into the matrix. So if you can imagine, like, the hydrogen would be the wind moving through the turbine, and as the wind moves through the turbines, it causes this to spin. And that spinning action is thanks to that kinetic energy of the hydrogen. So as the hydrogen is moving down its concentration gradient, as it's physically moving, it has kinetic energy in it, and we're going to take some of that kinetic energy. So think of it like water behind a dam. So we've got some stored potential energy up here, and then as the water moves through the generator, that generator is going to help create that ATP. So hydrogen is moving down its concentration gradient, and as it's moving down, it's turning the generator, which is your ATP synthase, and that ATP synthase is going to take ADP, an inorganic phosphate, and phosphorylate it into ATP. So because the NADH moved 3 hydrogens across the membrane, 3 separate hydrogens that are floating around are also going to come back in across the inner membrane through ATP synthase from the inner membrane space to the matrix. So our rule of thumb is, although this is not perfect, this doesn't happen all the time, 1 NADH is going to be equal to 3 ATP because of the 3 hydrogens that are coming across the membrane. And remember that in the Krebs cycle, we formed another coenzyme called FADH2. FADH2 is going to do the same thing. It's going to release electrons, but it's going to do it only starting at protein complex 2 and 3. And protein complex 2 isn't a transmembrane protein. So FADH2 will cause the third, which is cytochrome C reductase, to pump a hydrogen across. The electron moves on to the next one, pumps another hydrogen across, and then an electron is donated to another molecule of oxygen. So for FADH2, when it is oxidized back to FAD+, it's going to generate 2 hydrogens to move across the membrane. When those 2 hydrogens move across the membrane, 2 other hydrogens are going to move back from the inner membrane space to the mitochondria, and as they're doing, they're going to cause this generator to spin, and that's what's creating that ATP. So for every 1 FADH2, we get 2 ATP that are formed. All right, so what we've done is we've talked about oxidative phosphorylation, which is the process of ATP being produced as a hydrogen is moving down its concentration gradient from the inner membrane space to the mitochondria. We're using that energy of the movement energy to create ATP. But this process would not be possible if it weren't for that electron transport chain. So the electron transport chain's job is to create the hydrogen concentration difference, and it's going to do that by transporting an electron across multiple enzymes that are embedded in that inner membrane space. And with this, we are able to generate a large amount of ATP from a single glucose molecule. Thanks for watching.